DNA is very long and codes for all the activities that occur within a cell. To do this, it makes proteins to carry out the required functions for life. But how are the different types of proteins made? To understand that, let's first take a look at how DNA is replicated.
First, the DNA is unzipped by the helicase enzyme. As the DNA is being unzipped, free nucleotides in the nucleus attach themselves to the one on the parent strand by complementary base pairing. This is where adenine, shown as orange, attaches to thymine, yellow, and guanine, pink, attaches to cytosine, shown as purple. However, they don't just randomly come together. Hydrogen bonds must be formed between the nucleotides, and another enzyme called DNA polymerase catalyzes the reaction to make this happen. Each new strand always consists of half of the original parent strand as well as the new strand. In this way, DNA replication is a semi-conservative process because half of the pre-existing DNA molecule is always conserved.
Now that we understand what goes on in DNA replication, we can start on the process of creating proteins. The first step of protein synthesis is called transcription. This is when mRNA is created, which is short for messenger RNA. This is different from DNA in that the nucleotide thymine is replaced by uracil. As you can see, there is green where there should be yellow, representing uracil. Another difference is that DNA is a double strand, meaning that there are two separate chains of nucleotides held together by hydrogen bonds. RNA, on the other hand, is a single strand, so there's only one chain. The part of the DNA that has the code for the protein is unzipped by the RNA polymerase enzyme, similar to how the helicase enzyme separates DNA in DNA replication. Then, free nucleotides float in and attach to what is called the antisense strand. See that when mRNA is created, there is no thymine and instead there is uracil. Also note that RNA is a single strand, so only one side of the DNA is used for transcription. The antisense strand is the strand that is being used as a template for the RNA. The strand that isn't being used is the one being replicated and is called the sense strand. Once this happens, the resulting strand of mRNA is sent out into the cytoplasm through nuclear pores.
Translation happens when the mRNA aligns with the ribosome so that the first 6 bases are inside the ribosome. Every triplet of bases is called a codon and is essential as tRNA molecules bring specific anticodons to match the mRNA. An amino acid is also attached to it on the opposite end. Once the first codon has been matched, the second codon is matched with its own tRNA with its specific anticodon, and with it comes another amino acid. As you can see, the 2 amino acids are now lined up with each other. When an enzyme catalyzes a condensation reaction between them, a peptide bond is formed, and the 2 are at the beginning of what is called a polypeptide.
The first tRNA molecule then detaches, and so the second tRNA molecule shifts into the space that the first one previously occupied. This allows for a third tRNA molecule to float in, bringing yet another amino acid with it. This process continues until the final codon is reached, which signals for the end of translation. The resulting polypeptide is now ready to function, or it may need more time to continue folding and possibly combine with another one in order to be able to function.